Hello students, today we'll discuss denaturation and renaturation, right? First of all, I will start with denaturation, right? Denaturation is the phenomenon of disorganization of native protein structure, right? And denaturation results in loss of secondary, tertiary and quaternary structure of the protein. Only the primary structure remains intact, right? And there are certain agents that will cause denaturation. There are two types, that are physical agents and chemical agents, right? Now let's start with physical agents. Physical agents will include violent shaking, right? X-rays, UV rays and heat, right? And coming on to the chemical agents, which will include chemicals like strong acids, strong alkalis, right, or organic solvents like alcohol or ether. They will also act as chemical agents for denaturation, right? Now I'll start with characteristics of denaturation. First of all, the native helical structure of protein is lost and the primary structure remains intact, right? And denatured protein loses its biological activity on denaturation, and denatured protein becomes insoluble in solvent in which originally it was soluble, right? And on denaturation, the viscosity of the denatured protein increases and the surface tension decreases, right? And denatured protein is easily digested due to increased exposure of peptide bond to enzymes, right? And denaturation is usually irreversible process, right? That means it is not a reversible, but denaturation is irreversible, right? So it cannot be reversed. I will give you example of denaturation. For example, when we break the egg in the pan and make omelette out of it, it is example of denaturation, right? So omelette cannot be converted back to the egg, so it is a denaturation, right? But in some cases, careful denaturation can be reversed, and it is known as renaturation, right? And example of renaturation is, I will give you now. When you add salicylate to hemoglobin, it will undergo denaturation, right? But when you carefully remove the salicylate, it will regain its original structure, right? So it is an example of renaturation because it is reversible and hemoglobin regains its original structure, right? So careful denaturation is reversible in some cases. It is known as renaturation, right? But denaturation is totally irreversible. Thank you. Please like, subscribe and share.